Hello, everyone. I'm back. I figured it's been a long time since I actually did one of these presentations, and so I wanted to get into this discussion on aneuploidy and cell division. I actually had a difficult time coming up with the next topic of these discussions, and there's kind of this evolution of thought processes and building this foundation of understanding genetics. The previous one, we started out with a history of genetics, and then we went into kind of definitions of sexes. And what I wanted to do with this one is take a closer look at cell division, the various types of cell division, and what aneuploidy is all about and why it happens the way it does. So without too much introduction beyond that, we are going to take into the next slide, which is basically um, about me. I'm a board certified physician in internal medicine. I have no disclosures whatsoever. There's a couple of other things about me. I've been in practice for about 10 years plus now. And I really enjoy my work. So getting back to the crux of what genetics really kind of starts around is this basics of cell division, the central dogma, where we take DNA, we convert it to RNA, we take it to proteins. Now, there's variations in the midst of all of this, but this is kind of the starting point. This is the foundation of understanding genetics and how it works. We're kind of going back to the very beginning with this foundation of understanding DNA and how it replicates. And that's really where I wanted to start with this, is get back to the basics and understand what DNA chromosomes are all about and how they go from cell to cell and divide. The next slide is a little bit on the busy side, but I'm sure this is causing all sorts of medical students and biology students a little bit of a nightmare because we go into every single detail about each one of these proteins, what they do, the connections that are made between the ATCG um, nucleotides that are the basis of our DNA. What's really amazing with this is just the whole process. I'm going to see if I can get my mouse to work with this. Is when you have a DNA strand, this double helix, when it starts duplicating, it creates two strands. It gets broken up by this topoisomerase, which is demonstrated by this green ring. I'm sorry, this is the stabilizing molecule. The helicase comes in and actually starts splitting the DNA molecule. And it divides into two strands. There's a lagging strand and there's a leading strand. And the way this works is that the chain reaction that duplicates the chromosomes has to actually go in different ways. You can see that represented by the 3' and the 5', that they're flipped on each other. And they have to replicate the opposing strand in this whole entire process. So there's two different types of DNA polymerase. These are the molecules that start the process of getting these things combined, or the nucleotides back up and attached again. And they start linking each other up. And the easier one to do is this guy right down here with the DNA's polymerase. It basically just goes one right after another, a domino effect, just sequence, 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 sequence. However, if you look at the top one, you have this Okazaki fragment. Because the pathway of the lagging strand is different, you get these little fragments of nucleotides coming in and attaching to this DNA sequence, and they come in fragments. And along the way, this DNA ligase comes along and links them all back up together again. There's lots of videos. Actually, I took there's a video down at the link right here at genome.gov, which is a fantastic video. I highly recommend looking it over and watching this whole entire process. I know for a fact I probably did not do this process enough justice with that brief explanation. So I'm going to pop over to the next slide. In general, though, step one is you stabilize, unwind the DNA. Topoisomerase starts that process. You break the nucleotide links, which is the helicase, which is represented by that triangle, blue triangle right there. And then you start duplicating using polymerases to do that. And again, I represented that with a leading strand and a lagging strand. This whole process is incredibly quick, all things considered, considering all the billions of DNA sequences or nucleotides that we have to replicate. It can only take a couple of hours, really. However, if you were to stretch out the entire DNA sequence genome from point A to point B and start with one point and start to finish, it would take about 35 days. So obviously, if you're replicating DNA and you're doing it in a few hours, it's an extremely efficient process. What's amazing with this is that there's kind of this built-in error um, correction sequence. These DNA polymerases actually will, if a incorrect nucleotide tries to fit into its place, this DNA polymerase actually kind of unkinks it, kicks that guy out, and a new one comes into place to correct that. So in of itself, DNA replication is very accurate, but as with all things in nature, there are errors that do occur. Now, it can be as low as one error per 10 billion base pairs, which is incredibly amazing, all things considered. But in certain organisms, it can be as high as 1 in 1,000 base pairs. And one of the example organisms, or I guess objects you can look at with this, is the HIV virus has a high error rate. And as a consequence of that, it can alter the proteins, the antibodies that the antigens that our body's antibodies attack. And so it can hide really well from our body's immune system. And again, there's another link in there. I'll have this PowerPoint posted so that all these links will be accessible. So there are two different types of cell division. There's mitosis and meiosis. And yes, I did. I misspelled it right there. I didn't catch it because down at the bottom, meiosis is constriction of the pupil of the eye, and we see that with people who take opiates on a regular basis. But mitosis, and this is how we remember it in biology, is mitosis is what happens in my toes. You go from 46 chromosomes to 46 chromosomes. And meiosis is what occurs in the gametes, where you take 46 chromosomes, you divide it down into 23 chromosomes. So when you get down to the egg and the sperm, each one of them actually have only 23 chromosomes. When they combine to create an embryo, that's where you get the 46 chromosomes occurring again. Here is a diagram that demonstrates mitosis in general. It's an amazing process, and what's really neat about this process is obviously it happens all the time in our bodies. Even when we're awake, we're asleep. But as a cell divides, we start out with this interphase. And really what's happening with that is this whole start of the process. You can see these structures start to develop around the cell nucleus. These are what we call spindles, and they're creating these fibers to start attaching to the chromosomes. The cell nucleus starts to dissolve, and the chromosomes are lining up and unraveling themselves to the point where they're more easily accessible. This is early prophase. Late prophase is where things start to line up along the cell division line. And this is late prophase, metaphase. At this point, you can actually see in a microscope these phases occurring. As soon as you hit metaphase, all the chromosomes line up, the spindles attach to them, and they start pulling into opposite directions. And that's demonstrated during the anaphase. So they split in half and they go in different directions. As they get pulled in different directions, the cytokinesis process, the cells start to pinch off, and you have the chromosomes on one side and you have the chromosomes on the other. The cell membrane starts to form again, and those spindles and fibers start to dissipate again. And that would be telophase. And then we start back at interphase again, and the process starts over and over and over again. What's really interesting with this is, as you can see, there's a potential opportunities for errors to start in the midst of all this. Maybe one of those spindles doesn't capture one of those bits of chromosome, and it gets pulled in either direction. What would happen in that circumstance is the body would recognize that, or say that there's an insufficient DNA to replicate, and it would actually cause that cell to die. Meiosis happens in two phases. And this is what really amazes me with this is you have this DNA replication process just like we had before. Um, Cells get replicated and they enter in this meiosis phase. There is a certain part of the meiosis one phase where the chromosomes actually align with each other and they switch parts of their DNA. And this is part of the reason why we get our siblings looking different from each other, where someone can have curly hair, the other one can have straight hair, one can be taller than the other one, the other can be smaller. Or even if we look at it from a heritage ancestry standpoint, this is why we get variations in our heritage from different ancestries. For example, my grandmother is about 100% Norwegian, and as that went down, you would expect that my mother would be 50% and I would be about 25%. But if you look at it genetically, I'm actually only about 15 to 16% Norwegian based off of these DNA sequences. This is why. On the other hand, my sibling is a higher percentage than that. I think he's about 30 some odd percent, or just because of all these divisions. As we get into meiosis 2, what happens is the cells divide, but instead of pulling, um, they divide and they basically divide in half. They go from 46 down to 23. And that's how they stay. So the phase right here is what the ovary and the sperm look like. So this is representing only one chromosome. Here's that representation of chromosomal crossover. These chromosomes get close to each other and they break and they flip-flop. And you get these recombinant chromatids right down here, which are represented by the red and blue. Now, there's a couple of issues with this. As we analyze these passages and sequences that take place, we can see that there's a number of potential of errors that can creep into the picture here. There are problems with cell division. If you go back to any one of these cell divisions, you can see that these spindles can attach to a chromosome, and as they're working to split, they may or may not split, or they grab an extra one and pull it into the other cell. What that does is, for example, in meiosis 2, you can actually have an extra part of a chromosome that was not intended to be there. The same thing happens with mitosis, where the spindles, again, maybe instead of splitting each one of these chromosomes into two pieces, the chromosome doesn't break and the whole entire thing gets dragged over. And this is where we get the chromosome number being different than the 46. You can get 45, you can get 47, and you can actually get more numbers beyond that. And I'll have that represented here shortly. Chromosomes do break. You can get partial chromosomes that go through. Some of these sections can go missing. Chromosomes compare with non-analogous analogous chromosomes. And we can see that with the X and the Y chromosome trying to link up. You can take a part of the Y chromosome that carries the SRY gene, and it can break and go over to the X chromosome, which is unexpected. And as a consequence of that crossover, you can get a XX individual being born as a male because of the presence of that SRY gene. So even though there are fail-safes and checks in our cell division, there are errors that do occur. Aneuploidy is a variation in chromosome number. Typically, we associate it with the 45 and the 47 number of chromosomes, but it can be any number of aneuploidies. Most of the aneuploidies do not make it to term. There's too much DNA information lost, or it's uncontrolled regulation of cellular function. As a consequence of that, most of these do not survive. But the most common ones that we actually think of in terms of aneuploidies is number one, probably Down syndrome, which is trisomy 21, which means that there's 3 copies of the 21st chromosome. There's Klinefelter's, which is an individual who has XXY chromosomes. And then there's Turner syndrome, which is somebody who has just an X chromosome. They do not have an extra X or they do not have a Y. This next slide is very colorful, but what the most important thing with this is the non-autosomal chromosomes can have many duplications on themselves. What's interesting is that in individual who are embryo that has just a Y chromosome, in other words, they do not have an X, is not a viable term or a viable pregnancy, and it will automatically terminate. There's way too much information on our X chromosome that we actually need to have that must be carried forward. Otherwise, we just can't survive. So the X chromosome is absolutely vital to our survival. Now, as you can see, there's lots of variations with this. There's XXY, there's XYY, XXYY, X naught, and there's interesting things with this. So if you look at the XXY individual, 2X's and a Y, the extra chromosome in of itself can come from either parent. As a sperm can have an X and a Y because the male individual can have an X and a Y show up in the sperm. The extra X can also come from mom and work that way as well. So in terms of Klinefelters, both parents could be potentially culprit behind either one. The X Turner syndrome can be either as well. Interestingly enough, with Klinefelter syndrome, actually, I'm getting ahead of myself. I'm going to actually take into the next slide as with the autosomal. This is a little bit of a busy slide, but the reason why I bring this one up is to show all the aneuploidies associated with our genome. To the left, you see the chromosome number all the way up through 22, recognizing that the chromosome 23 is your X and your Y chromosome. The next part of the table, the monosomy, is basically one of the chromosomes is missing. And you see that with one of the chromosomes missing, you still have viable pregnancies, viable terms. When you get to the trisomy where there's 3 of them, most individuals do not survive with the trisomy. The one that we are most familiar with is the Down syndrome, and there's only a couple more. Those that are in the red category are not viable. The ones in the yellow are potentially viable, though they do have significant genetic abnormalities at birth. Down syndrome tends to be more likely to survive. So I wanted to take a step back and talk about individuals who have Klinefelter syndrome. Again, these are individuals with 47 chromosomes, XXY. The incidence of this is about 1 in 500 males. Now, one of the things I teach my medical students is you have this foundation of the textbook. There's always this textbook answer, but I always teach them that there's always variations away from the textbook. You can also think about this with working with musical instruments, with performing an art, that there's the basis of our understanding, the foundation of our knowledge. But as we get more experienced and more practice in our specialties, that we realize that there's variations on these themes. So when I write about this, they typically will be tall. They may have gynecomastia and they may have difficulties with fertility. They do not have to be. And what's interesting is if you look at this individual over to the right, you would not actually stand out, this individual would not stand out as somebody who has specifically Klinefelters. They look very typical male. In this case, they do appear to be relatively tall. They may or may not have gynecomastia, which is male breast growth. That may or may not be the reality of it. Now, typically, these people are assigned male at birth. However, there are a few cases out there where the genitalia are ambiguous, and that is a reality that's written in multiple case reports. Treatment can involve hormone replacement therapy. Depends if they need it or not. Maybe they have sufficient testosterone that they don't need that. Maybe if they do develop gynecomastia, one of the things that's offered to them is surgical removal of the male breast tissue. What's also interesting with these individuals is gender dysphoria is also present at a higher rate than the general population in Klinefelter syndrome, which, if you think about it, they have an extra X in their chromosomes, even though they are assigned male, typically assigned male at birth. That extra X does play a role in our development, as we've talked about in our previous lectures on the sexes. Turner syndrome is 45X naught and occurs in about 1 in 2,000 females. Interestingly enough, this is a interesting statistic, is 10% of spontaneous abortions in the United States are due to Turners. Think of the vast majority of the spontaneous abortions that happen, and to recognize that 10% of them is because they're missing that second X. That's an amazing number and shows the reality of how frequently this actually does occur. Again, going to that's a similar theme to Klinefelter's. In general, Turner individuals' appearance is also incredibly variable. They can get what's called a webbed neck, where their neck looks a little bit wider and tends to have a skin fold that's part of that. They're typically shorter in stature. They do typically have congenital heart disease, and it's not uncommon for them to have other birth defects. What's also interesting with this is that these individuals can be fertile, even though that they're missing one of the X chromosomes. If you look at this Punnett square, you can see that if you have a Turner's patient married to a male, this is what happens. There's a 1 in 4 chance that they have a female at birth, or a female assigned at birth. A male assigned at birth is 1 in a 4 chance. There's a 1 in 4 chance that they have a Klinefelter's, or not a Turner's individual at birth. And then there's a 1 in 4 chance that the pregnancy is not viable. So there's usually a lot of discussions with these individuals about fertility and risks and genetic risk and things like that. So I hope that that presentation was helpful. Here are my resources. I have others that were listed in the lecture as links. This one actually references the gender identity and questioning with Klinefelter syndrome, and demonstrating that gender dysphoria is more common with Klinefelter's patients. The aneuploidy, I got this diagram directly from Wikipedia, which was helpful. And I went back to errors in DNA replication was an article from Nature. And then I did pull in a DNA replication video from genome.gov, which is an amazing video process that is well worth watching. So again, thank you for your time, and there'll be more videos to come. Have a good night.